Okay, well, good evening. Welcome to Planet Ocean. This is our third presentation of the season. We actually have two more. We have one more that's scheduled and a fifth one because we missed last year. Yeah, so we're making up for lost money, time. And that, so the last one of the season after the next one, I don't recall who all this is. Carms is tonight, but is Rob Condon is a new faculty member in biology. We talk about jellyfish, and so that'll be kind of a new thing for us. Tonight, it's my pleasure to introduce my longtime friend, probably since the late 70s when Carm and I first met each other. I'm trying to read this as I talk to you. I just got it. Carm came back to the United States after getting his PhD at University of Rhode Island. He was in Italy for a number of years, doing fine work there in phytoplankton culture collection. Came back to the States to the Florida Department of Natural Resources. It's not called that anymore. Florida Marine Research.
Yeah, WC.
Something like that. But anyway, came back and worked with the renowned Karen Steininger, and Carm actually carried out and developed the culture collections in Florida, more than, I think it was bordering on 1,000 different species and clones, many of which he brought here to UNCW when he came. About midway in early 2000, there was a series of fish kills up and down the coast, Delaware and mid-United States, that were rather peculiar and resulted in ultimately the identification of a new organism that Carl will talk about and a specific group of toxins that related to other fish-killing toxins in the United States. This is a real sleuth story. I'm sure he has some interesting stories to tell about it, and I'm going to ask my dear friend Carm Tomas, one of the members of the Marbionic trio that came here from Florida, and from, where did I come from? I came from the University of Miami. Carm was in the west coast of Florida, and Jeff Wright was in Canada. And for literally 20 years when we would get together, we would talk about trying to find a place where the three of us could get together to actually execute on our vision for the marine sciences and archaeology. And what you see around you is part of that. So, without further ado, my good friend, Professor Chatenella Marina.
Thank you. Well, good evening, everyone. This Thursday is obviously a lot better. Tuesday is obviously a lot better than last Tuesday, and I'm glad that you were able to make it tonight. I have a scientific mystery story to tell you. It has all of the elements that the CSI things on TV have. It also has some other little interesting twists. So as I sort of introduce this topic, I just want to tell you that this event has an unknown serial killer. It has killed thousands, if not millions, of individuals. Rumors galore occurred about this this event when we were trying to figure out what was going on. Everything from people getting sick just by being at the seashore, to having epileptic fits, to having respiratory distress, and there was even a mention of a possible CIA involvement. Talk about that. But the real story is about the high-tech series of investigations that we did to try to figure out what was going on. In these investigative tools are things like an electron microscope that magnifies things hundreds of thousands of times, something called an HPLC, high-performance liquid chromatography unit that describes all of the different pigments that this organism has. We did something called two-dimensional Nmr spectroscopy, ROSI as it's called, to identify a special compound. And of course, there had to be DNA fingerprinting in all of this. This is all part of the story. And then to end it off, we did some toxin analyses in conjunction with Dan's group in trying to identify what was going on. And again, using a whole series of very specialized techniques. And so as we go through this, I'll try to make a lot of this intelligible for you folks that maybe aren't familiar with a lot of these things. But it was one of these things that was presented to us. It fell in our lap, and you had to decide to do something with it. And the thing we decided to do was to try to pursue it to the point where we could answer some of these questions. Of course, all of this takes place in Delaware, of all places. Poor little old Delaware. And not only that, but in a special region of Delaware, where there had been a series of unexplained fish kills during a summer, 10 of them in fact, that again were a mystery to everyone, and they were trying to figure out what was going on. So let me just turn the lights down a little bit so you folks can see a little bit better. Second from the bottom, I believe. That one. Okay, there we go. And so this image that I have here is actually a volunteer. He was the president of something called the Surf Riders Association. Some of you may have heard about it. The Surf Riders Association is a group of people who are interested in surfing, but they're also very, very much interested in the environment. And the people in Delaware were particularly concerned about these fish kills that were just happening, and no one seemed to have an answer. And they were really worried that the Department of Natural Resources, DNREC as it's called in Delaware, was really kind of either not paying very strong attention to it or maybe not telling the truth about it. And so they were interested in getting other people involved. We got involved by an indirect method, and this is my mentioning of the CIA, and I'll mention that briefly to you. But it was, again, one of the events that we expected. Now, which one is this, Dan? The one on the right? Okay. One of the reasons why I got involved is that I've been studying for many years these little organisms. These are one-celled organisms called phytoplankton. They grow in the ocean. These happen to be color-coded. There's green ones and there's brown ones. The green ones are freshwater ones. The brown ones are marine. Of course, this one is colored by various kinds of dyes. But in any case, these organisms are modal. They move in the water column. They can swim 30 feet in 24 hours up and down the water column, and oftentimes bloom or create these blooms, what we call blooms, very, very dense populations. And so the kind of thing that happened in Delaware turned out to be something that was very similar in appearance to this particular kind of organism. Now, where this all takes place, this is the eastern seaboard, as you know. These are the two extremes of the known world. There's Wilmington, North Carolina, New York City. All of this happens in this little area right here called the Delaware Inland Bays. That's a rather important area because it's really accessible to people from Maryland, Virginia, Washington, D.C. as an area which is very desirable as, again, a vacation area. Again, here's Delaware Bay, the large bay. Here are the inland bays down here, and I'll show you a little better look of the inland bays. Here they are here, where there's a very, very narrow opening to the Atlantic. So there's very little seawater that gets through there. But what happens is that all of these bays in through here are really sort of places where we say that the things can cook. Okay, the outer banks are like this also because it's actually an enclosed embayment of some kind where things that are being released into the water can stay there for a certain amount of time for the organisms to take care of them. And what is there around here besides the beautiful vacation homes? But this is a rather agricultural part of Delaware where they're the counterpart to us. We have more hogs than humans in North Carolina. They had more chickens than humans in this, and many of them were in this particular region. But the areas of particular interest is this thing called Bald Eagle Creek, Torquay Canal, Russell Canal. This section up here, this is where the kills, the fish kills were first noticed. And again, we got samples from a variety of other places along here. Now, if you look at this area, this is a typical Atlantic estuary, beautiful salt marsh grasses. Again, very, very desirable place to spend some time. Rather very nice homes. Again, an area that is well characterized. In August of 2000, this happened. These are dead menhaden. Menhaden, those of you that don't know them, are an oily fish. You probably know menhaden by, if you take omega-3 capsules, and especially if you say, oh, these taste fishy, that's because they're the oils that oftentimes come from menhaden. They're pressed out of the organisms. But in this instance here, we have about 2 million dead fish. There's a turtle here. There's also dead blue crabs, dead clams of various kinds, oysters. And to add insult to injury, this happened right in front of a yacht club. So, you know, people really got concerned about this and said, we got to do something about this and what was going on. And why are we having these events? And so we asked for some samples, if we could get some samples, after getting a tantalizing sample that came to us indirectly. Now, the tantalizing sample came from this person here, Mr. Winkler, that runs a small shop at Bethany Beach. His concern was that people were reporting that they were getting sick, that they were having spasms, that they were coughing and wheezing from being at by the water, and that something was happening, and the DENREC was not coming clean. They were not saying anything about this because they didn't know what was going on. There was rumors at that time, whether it was Mr. Winkler or not, I'm not sure, that maybe the CIA was even releasing nerve gases, or getting rid of nerve gases offshore. This is not the first time that's come out. When we worked in Florida, it was the same thing off of the east coast of Florida when they had fish kills. But in any case, we received a sample via Woods Hole, Massachusetts. This sample went to Woods Hole, stayed in a refrigerator for 2 weeks in the dark, in the cold. This is an August sample. And by the time we got it, there wasn't anything that I could recognize inside it. And so we said, no, we've got to see something a little bit more, more realistic, more close to the real organism. And we made contacts with folks in Delaware, and eventually the folks from Surfrider got in touch with us. And they said, we're having a fish kill now. We'll collect some samples right below the fish, and we'll send them to you. And that's what happened. And they did send them to us. Eventually, DENREC did give us some samples that they had archived, that they had preserved. However, the real samples were primarily from Surfrider, as well as some samples that we took. And I'll tell you about the archive samples in a minute. What we were seeing in these samples, and these are samples from August through October of 2000, is somewhere in the vicinity of 1 million cells per liter. Now, a liter is just a little bit larger than a quart of water. All right, so and in fact, on August 28th in Torquay Canal, where there was this big fish kill, there were 10.4 million cells per liter. So there was a lot of material there. The interesting thing is that these samples were sent to 5 different laboratories. I won't mention, out of courtesy to my colleagues, who they were, but they were all sent to these other laboratories, and time after time after time, we received word that they didn't see anything unusual. There was hardly anything noticeable to them. There's a reason for that, and I'll explain shortly. But anyhow, the organism that, that I was looking at was this little round thing. It looks like a small tennis ball, clustered green chloroplasts in the center, almost clear ring around the outside, two flagella. Flagella, by the way, are these whip-like structures that they wave in the water, and that allows them to move through the water, and I'll show you this in a moment. And again, it was something that was a bit unusual, not like anything that I had really seen except in the literature. However, looking at them a little bit more, there were these slipper-shaped cells, heart-shaped cells, but the amazing thing about them is that these green clustered things are chloroplasts. That's where the chlorophyll is contained in the cell. And it seemed like they were bulging. They were bulging out of these cells and almost looked like a cluster of grapes. And which then said to me, well, boy, if I ever name this thing, if it's a new thing, I have to have the word grape in it somewhere, because that's what it looks like. The Japanese had published this thing, which they call Chatean Olivericulosa. This is an organism, a phytoplankton species that blooms in the inland sea of Japan, where they have many, many aquaculture pens, where they're raising things like yellowtail, which is a fish that they use primarily for sushi. And this Chatenella Marina had caused large fish kills. And I said, you know, strangely enough, it looks close to that, but what is a Japanese species doing in Delaware? You never know, but in any case, it didn't quite look right, but it was probably the closest we had. So what we eventually did was I call this species, and this is a picture of the bloom here, and this is a different species here, but all of these smaller green ones with the tiny tails are the organisms that I'm talking about. And I called it Chatenella CF Vericulosa. Now, if you don't know what the real thing is, we have certain hedge words that we can get away with. CF means it looks like, but it's not exactly. In other words, we didn't identify it as that particular organism. And so what we had then is the CF Vericulosa. And I want to show you, I'm hoping this will work. I want to show you what this thing looks like. This is the actual sample, and this is how these guys move. And you can see them. I don't know how you could look at the sample and say there was nothing in it. The reason why they didn't see anything in it was because most of their samples were preserved. So that when they collected the samples, they put some kind of preservative which killed everything, turned them into raisins, which are indistinguishable at that point, and so they were no longer something that they could see. In the meantime, we looked at this and we said, oh, wow, what is that? Who does it belong to? What does it look like? What's going on here? Okay, so that is a picture of the actual bloom during the 2000 thing. Now, I have a colleague in Japan, and his name is Yoshimatsu, and I said, do you have any images of Chatenella Vericulosa? And he says, oh yeah. He says, let me show you this one. Now, making corrections for the difference in photography. This is what the Japanese organism looks like. The first thing you'll notice is that it's not green, but it's brown, golden brown. It has a slipper shape. It has that same wrinkly covering, but it's slipper shaped, but it's still not very convincing that it's the same organism at all. So after seeing this, I said, no, it can't be that. It's got to be something completely different, or at least from our observations, we needed to look in a different direction. And so we had at least a lead, but we didn't have anything that was more definite than that. One of the other things that came along was this wrinkly looking long thing. Now again, it's about the same size as the cells that we were looking, even though it looks much longer than this. And this is something that was found in Norwegian and Swedish coastal waters, where their salmon were dying. They were being killed by this ugly-looking caterpillar-like thing, and they call that one Chatenella Verriculosa. See, they played the same game as I did. In other words, they said it has an affinity to verriculosa, but we're really not sure it's the same one. And so this one has since been renamed. It's been called Pseudo-Chatenella, and it's basically put into the same group that the Japanese one was, but in a totally different class. Okay. All right, so first of all then, we're going to take close looks at this organism, see what it looks like. And here, these are drawings, and our illustrator, who is sitting back there at the video, I think she's back there. Yeah, she is. Anyhow, she drew us these beautiful pictures here. This is what the organism looks like in line drawing. It has all these chloroplasts surrounding a central nucleus, and then it's got kind of an empty area and has a long tail as the slipper-shaped one that is transparent. And here's a picture. Now here's some oval ones, but they can also be completely round and again be pretty much with clear areas all the way around them, but the chloroplast within the center, and two flagella. Okay, two flagella. Now these images here are with a scanning electron microscope. All right, and so these again are close to 100 times their magnification. And what you can see, the important thing here is that they have no covering on the outside. They don't have any kind of scales or plates or things that we find in other species. It's just like it has a clear cellophane wrapper around it. And it has two flagella, as I mentioned to you. One of them has little hairs that allows it to grasp the water. And these flagella work one way. The forward one wiggles back and forth, actually getting traction in the water, while the second one is trailed alongside of the cell. So the front one serves as the propeller, and the one that's hanging down is the rudder. So it can orient itself. And then, of course, we found an opening which is quite unusual. But again, you can see these bulging chloroplasts. It's rather unusual. Now, this one is something called a transmission electron microscope. In this one, we take these cells, fix them, slice them very, very thinly, and then look through these slices. And some of the things you can see, here's the flagellum with the hairs on it, for instance. We can see this part. This is the front end of the cell where the flagellum emerges from the cell. It's a very important part of the cell because the structure in here can tell us a great deal. It's not like any of the other Rhaphtophytes that I normally study. So it's not a Rhaphtophyte. These are chloroplasts. Then you can see that they're emerging here. This is the nucleus with the nuclear membrane, mitochondria. These are all organelles inside single cells that do different kinds of jobs in surviving the survival of the cell. Okay, so we're looking at the insides of this cell, and we're saying, you know, again, we find things that are found in other organisms. However, it's still pretty unusual. We're still not sure where it belongs. All right, it took us about 3 years to get it into culture. One of the things that we do here in Marbionic and at the center is to be able to get these cells, get them to grow, and actually get them to maintain them in culture so that we can grow them up in large quantities and do whatever we need to do with them. And so this is what it looks like in culture. It forms these greenish strands. However, for the most part, they're single cells. And again, there's another image here of individual cells. And you notice, again, you have very clear areas all the way around with just some green towards the center. These strands, by the way, are just sort of tubes, clear tubes where individual cells are inside. There's not anything other than that in there. All right. Now, in looking at this organism, we kept on saying, well, what kind of conditions does it grow in? And So we took a little clue from nature where we found them. And this is how you proceed with some of these things. When you're starting to grow a plant that you've never seen before, you need to know where it comes from, what conditions it grew under, and so forth. And so here we have, again, this is a summer-fall species. After having a couple of years of observations, we could say, well, all right, it likes temperatures around 22 degrees centigrade, so that's fairly warm. It likes longer days, so that you would expect, for instance, in mid to late summer. It has the kind of nutrients that were found in the water, or the standard nutrients, nitrate, ammonia, phosphate, and silicate. But the ratios of nitrogen to phosphorus told us that they would run out of nitrogen before they would run out of phosphorus. So nitrogen could be limiting. And they were accompanied by this group that I've studied for many years, the Phytoplankton in 2 species, Chatenella subsalsa, heterosigma cashua, another dinoflagellate, Tribidinium striatum, and then some euglenas, which are, again, are green algal cells, modal single green algal cells. Then using a very simple system of a circulating water bath and a Turner design fluorometer, we did some growth studies. We tried to see what would get these things to grow. And again, this is what we found. With salinity, for instance. Now, full strength seawater is 35. All right, 0, of course, is distilled water. And basically, in trying to grow this at different salinities and stepping it up by 5 units of 5, we found that this species liked to grow at 15 to 20, all right, but then tapered off at either end. That told us that this was a species that likes to be in estuaries. It likes to be in places where the seawater was diluted some, but it wasn't really full fresh water, and it wasn't full seawater. Although it could survive 10 and even 30, its best response was in the intermediate salinity. Now, this next series, we started looking at nitrogen, and we said, you know, how does this thing grow? And this is what we can do in culture and with these, with that fluorometer, that machine that measures chlorophyll fluorescence. And again, so we have days here, and this is the relative fluorescence, the amount of fluorescence that we would get. And obviously, the more fluorescence, the more cells that are there. And so when we get these kind of peaks here that are increasing, we're basically seeing a response that the cells are growing. And here what we did was we looked at ammonia. We said, well, you know, ammonia is one of the major nitrogen sources. It turns out that ammonia was considered to be the preferred nitrogen source because it becomes, it enters the cells and then it becomes amino acids, part of amino acids, very, very quickly, without doing very much chemical switching. And so what happened was that as we increase the amount of ammonia, you can see in the black triangles, we actually got a diminishing growth. And so it likes ammonia, but not that much. And if we got to a point where we had 30 micromoles of ammonia, which is not unusual for coastal waters, it didn't grow as well as if it had less than that amount of ammonia. All right, we then tried nitrate and ammonia in this dark circles, but look at what happens when we use urea. Now, urea, you have to understand, is a detoxified form of ammonia. I mean, animals, we produce urea because ammonia is really toxic to us. And so in that process, it produces a byproduct which is less toxic, which is a little bit more complicated. But on the other hand, for plants, it's a very, very important nitrogen source. In fact, urea now is used as the major nitrogen source in agriculture. Why? Because it tends to stick around for a while. It doesn't just flush out. It takes a while. It's refractal. It takes a while for it to be used up and to break down. Okay, so here's this organism that's using urea. Remember I said something about chickens? What's the waste product of chickens? Urea. Okay, so it fit kind of nicely in this whole story, but you know, it's just sort of an indication. Now, what I'm trying to tell you is show you how we build on the information, step by step by step, by getting a little bit more and a little bit more information about this organism. Okay, now if we look at urea and ammonia combinations, or urea alone, urea and ammonia, as we add ammonia, it doesn't grow as well. So there is a very clear indication that it likes that as a source, a preferred source. And so if we look at the different nitrogen sources, and remember this, from the amounts that we could tell, it was going to run out of nitrogen first before it would run out of anything else. And if we look at nitrate, ammonia, and urea, for this CF auriculosa, it grew quite well with urea, whereas some of the other things it really didn't care. In fact, this one grew with ammonia quite well, this Heterosigma acasio. With Gyrodinium instriatum, it did not like ammonia at all, and again, it grew well with nitrate and urea. Why do we do these experiments? Well, we've got these organisms in the same environment. They're competing for the same resources. They're looking for some edge to be able to survive, some edge to be able to form these blooms. And our question is, what is that edge? What is that thing? And in this instance, it looks like that the nitrogen source plays a very important part in who's going to win when you've got a bunch of organisms that need nitrogen and the kind of nitrogen that's available. Okay, now we're going to switch gears a little bit and talk about high-performance liquid chromatography. So I know you're all sitting on the edge of your seats about this one. What is high-performance liquid chromatography? Well, it's this little machine here. It costs about, what, $60,000, something like that. Anyhow, it's a machine that we have in the lab that if we take a sample and take all the cells out onto a filter, extract that filter with something like methanol, okay, methyl alcohol, and then put it through this liquid chromatography unit, we get this. And each one of these peaks are the different pigments that this machine has been able to separate out. So we find out that in this sample, for instance, we have 8 pigments, and again, they're sort of distributed this way. This is called the retention time. It means what time it takes for that pigment to come through the machine and go by the detector, so it makes that indication. And of course, the height is the amount. So there's a lot more of pigment number 7 than there is, obviously, of pigment number 2. All right, let's take a look at this. And now, this is the spectrum from the cultures of the unknown species. This is from our cultures now that we grew. And again, here's the retention time. And we've now labeled the different pigments. So we can tell you that this one here, the biggest one, is chlorophyll A. All photosynthetic organisms have chlorophyll A. Okay? All terrestrial plants have chlorophyll A. But the interesting thing about this species, it has another kind of chlorophyll, something called chlorophyll C2. But then it has heteroxanthin, diadeinoxanthin, diatoxanthin, zeaxanthin, beta-carotene. You know about beta-carotene. This is something that's good for your eyes, and it's also part of supplements. Many people take that. And so you see all of this. Now, those of you that are really astute will notice that I skipped one. Yeah, there's one there. That's right. And that one is this one right here. It had this unusual pigment. And we're trying to figure out what on earth is that pigment. We don't know what that is. And we have a way of taking that pigment as it comes out of the machine, capturing it, and then looking at some of its characteristics, its absorption spectrum. And this absorption spectrum, which is, again, that pigment and the peaks of that pigment at different wavelengths, was a typical carotenoid pigment. It belongs to the same category as heteroxanthin, diadeноxanthin, beta-carotene, but it's not any of those. So he said, what on earth is going on here? All right, so we have a new pigment, something we couldn't identify. This is the Hab species that we're just talking about. All right, this CF verruculosa. And these are the different kinds of pigments it has that we could find in it. And in this case, we made them equal. So we divided them by the amount of chlorophyll. So if one had twice as much chlorophyll as the other, we canceled that difference out. So we could just compare the composition of the pigments. And here we can see it has diadeноxanthin, 47%. It has about 32% of the unknown. It has some fucoxanthin. It has diazoxanthin. So again, it has a beta-carotene. It has a number of those pigments. Now, what we did was we ran some of the other species that we thought it was like. Here's Chatenella Marina, the one from Japan. Remember that I showed you before in that drawing? And here's something called Chatenella Marina, another species that forms a slipper-shaped cell. It's golden brown. It, again, is oftentimes associated with fish gills. And these belong to this group called the Rhaphtophytes. Well, when we looked at the pigment profiles, we discovered that this one had chlorophyll C3. The other ones didn't. Okay, so that right away told us it was not a Rhaphtophyte. It belonged to something else. And indeed, what happened is that it is now put in a totally different class called the Dictyocophytes. And so here's how the pigment profiles can help us in being able to tell us what the differences are. But we still had other relatives, we thought, that would be worthwhile looking at. There's a group called the Xanthophytes, and the Xanthophytes have a particular a series of pigments, and we decided to take a look at those because they might have given us some indication. And here's what we find. We find that the Hab species has this diadenoxanthin, and all of the Xanthophytes do. It still has this unknown peak that's not in any of these others. It has this thing called Valcherioxanthin, which is here. And there's also a little bit of something that looks like Valcherioxanthin here. But then heteroxanthin is another one. And so there are some differences, but it doesn't really match. It doesn't match well. There's still this nagging thing about 32% of this unknown pigment. And what is it? Where is it from? And this is where some colleagues of mine in Italy, who are masters at this kind of work, used something called two-dimensional NMR spectroscopy. This is called ROSI. Why do they call it ROSI? You know? Pardon me?
Because the Italian words for those things?
No, I don't think so. But it's two-dimensional. Pardon me?
The red spots.
The red spots? No, it apparently because there are different versions of this. But in any case, what this does is it gives basically a signature that allowed my colleagues to identify the structure of this pigment, to actually tell us what that molecule looked like. And this is what happened. Now, the Xanthophytes have this thing called Valcherioxanthin. If you look at it, this is this complicated pigment. And again, at all of these junctures, you have carbon atoms, and then you've got hydrogens and hydroxyl groups and so forth. And so you get this kind of structure. This is what they discovered with the new pigment. It's very, very similar. Okay, so it's perhaps a derivative of Valcherioxanthin. But notice where the differences are. The difference is this group right here, and there are some differences. This is a subtle group right over here. And so this became a brand new pigment. A brand new pigment that was found only in this organism. Okay? It's found only in this organism. And so we gave it a name, and I'll tell you something called mormxanthin. But in any case, it's typical of this particular organism. Incidentally, what we had to do is we had to sit by the HPLC, and whenever that unknown peak came off, we had to put tubes underneath the exhaust of this thing to collect that peak. We did this enough times until we got enough material that we could then put it on dry ice and send it over to Italy for my colleagues to work. This was kind of an interesting thing in itself, because we put something like 25 or 30 pounds of dry ice in a box. And when it arrived in Naples, because it got stuck in customs in Milan, it arrived with like 10 pounds. And fortunately, it was still frozen, and they could use it, and they were able to get the structure. Okay, so this is the natural sample from Delaware. Oh, there's something about scientists. They're crazy scientists. They never throw anything away. And also, you try to think about what's going to happen ahead. And so when we had that bloom in 2000, we took and we filtered some samples, and some of them we put in a -80 freezer, which is one of the freezers that we use that allows this material to be very, very stable. This is now 4 or 5 years later. We take out those same filters, put them in methanol, extract the pigments, run them through the HPLC, and see what we get. And sure enough, here's the tracing that we get. And the interesting thing is that there's that peak in the natural sample that we got in the culture. And so this pigment, mormxanthin, is unique to this particular organism. It can be used as a tracer. Even if we didn't know that it was there, if we could run HPLCs on pigment profiles on samples, and we found it, then we could say it's absolutely there. Okay? And in fact, someone did HPLC analyses in Chesapeake Bay, Solomon Island Laboratory, and going back in their archive data, they were able to find that same peak. So they had these organisms at some time in their history. Okay. Okay. Now I want you to memorize this table. No, the thing is that this is the CF verruculosa that I've given another name to. These are the pigments that we have, and you can see that with all of some of the other species, the Xanthophytes, the Rhapidophytes, the Dictyocophytes, that they share a number of the same pigments. There are some that are absolutely missing, like this group right in here, but the one that's unique is this mormxanthin. It doesn't occur in anything else. Okay, so I've beaten you over the head with that. All right, so now we get to the genetic fingerprinting. Well, we have been taught some very interesting lessons about genetic sequencing. And we can identify, for instance, just from droplets of blood, for instance, where that came from, or if we find a match with an individual. And the problem is that it's very, very difficult to do that, to do it clearly, to do it consistently. But you also have to have a library that you can go back and say, oh, it matches up with this one. Okay. And so part of the difficulty we discovered was that the genetic sequences that we were doing were not leading us anywhere. Well, it wasn't quite a Rhapidophyte, and it wasn't quite a Xanthophyte, and it wasn't quite something else. So it was very frustrating. And you know, what I wanted to do is I wanted to go to my molecular colleagues and say, lead me out of the forest, take me by the hand, and show me exactly what it is, because I mean, I still don't know what it is after all of this. We know that it's something unique. It has a unique pigment. It has unique form. But you know, where does it belong? What group does it belong to? All right, so what we did was we looked at different genes. This is a small subunit of the 18S subunit of ribosomal RNA. We have the ITS spacers, which again is a different part of that gene. And we looked at this group of clones, but then we also looked at other genes. We looked at genes from the large subunit. We looked at genes from mitochondria, COX genes. We looked at genes from chloroplasts. All because somebody was saying, well, you just can't look at those, you've got to look at others. Okay, so 18S nuclear encoded RNA sequences. We got those sequences, and there isn't much to show you as far as what those are concerned, is that you get these sequences, and then you go to something called GenBank, where all of these sequences have been deposited, and you run a search to find out what in there is the same or similar to that. And so we did that and ended up using something called an ARB analysis, which is a secondary structure. It does more than just the linear kinds of genes. It also how these genes are folded. But we also did the large subunit, the D1, D2 region, this plastid encoded gene, this mitochondrial gene, and then we built these trees. Now these trees supposedly are statistical trees that are built to tell us what is like what, and how much are they like one another. Okay, and so here we have, now all of these are cultures that we isolated from Delaware. So we have 8 of them. And they all cluster together very nicely. There's 100% similarity between all of them. But it really doesn't fit very well with these others, because these are the Rhaphidiophytes over here. And these are some Chrysophytes over here. And at this point, it stands out alone. And so we're saying, darn, there it is again. It's just not telling us anything. Actually, it was telling us a lot, but it's because of the way you're looking at this thing. You're saying, tell me something. Here's another analysis, and here it is up here. Here is Chloromorum up here. And again, this is the secondary structure. This is the more complicated one. And again, it has very, very good agreement among the individuals there, but it has absolutely no agreement with anything else. So it sort of sits out there by itself. And I'm saying, well, where are the other things? Where are the Xanthophytes? Here are the Xanthophytes over here. And I'm saying, well, where are the Rhaphidiophytes? The Rhaphidiophytes are close, but they're not within that same group. Okay? Now we're looking at the large subunit, D1, D2 region again. And again, very, very, these are called bootstraps, very, very poor bootstraps, sitting out there by itself. And again, when we look at the plastid encoded genes, we have it down here with very, very poor connection to anything else. And this is the COX gene, this is the one, the mitochondrial gene, and here we have it also in here. And again, it has very, very poor support with any of the others. So time and time and time and time again, there's this old saying about how many times you beat yourself over the head with a hammer before you realize the hammer's harder than your head. This is telling us it's different. It's not like anything in there. Okay, and will you please understand that? And so, and there, if we put all the genes together and build another tree, we just end up with the same story. All right, so what we basically have come up with is the fact that this is basically a new not only a new species, a new genus, okay, but it's a new class, which puts it above all of those other normal definitions. Now, new species are found relatively commonly. New genera are found less common, but again, it's not unheard of. New classes are maybe found once every 10 years or more. And so here we have something which clearly tells us it's a new class. The name that we've given it is Chloromorum toxicum, okay? Chloro meaning green, morum meaning mulberry, toxicum, of course, being toxic. You have to pick a unique name, a name that wasn't used before, or else it's not accepted by the botanical code. And so we took this one. I wanted uva formis or something like this, which meant in the shape of grapes, but that's been used many, many times. Okay, so the new class is the Chloromorphiceae, the Chloromorales, the order, the family Chloromorphiceae, the genus Chloromorum, and the species is Toxicum. All right, so we have that. Up to this point, then, we've identified this as a totally new organism. Totally new class, new genus, new species. All right, now, as far as the toxins are concerned, we have several lines of evidence, and this is one of the very first ones that was done in conjunction with Dan Baden's lab, Andrea Bordelais here, who's here. Basically, this is, again, a time of when there was a bloom in Rehoboth Bay. If we take the bloom water and use chloroform and extract the bloom water, we can separate the chloroform from the water, we can dry it, and then using different solvents, clean it up, and then using a chromatography system like the one for pigments, but this one specialized for the pure compounds of the toxins, be able to separate the toxins out. And we ended up with this interesting thing, which we published, by the way. And here again, in this particular profile, we find that there are peaks of the toxins that were well-known peaks to our people here. That these are really brevetoxin peaks, peaks of toxin from the species in Florida that produces the Florida red tide. And so we have PBTX3, 9, 2, again, which again in small amounts, but they were there, that were present in the HPLC tracing. Now, using a totally separate system, something called an ELISA, enzyme-linked immunosorbent assay, and comparing the responses of the different peaks and looking at all of these peaks, we found out that the ELISA showed us that wherever we saw something that was looking like a brevetoxin peak, the ELISA was saying, yes, it's a brevetoxin, or it's responding like a brevetoxin. Okay, so we have very strong indications that the Delaware sample had at least these three toxins. PBTX2 was the one that was most common and was identified in the highest concentration. The others are in lesser concentration. The other evidence, again, the other chemical evidence is something called mass spectrometry, where again you use a specialized instrument to be able to look at the different components of those toxins. This is a brevetoxin standard, and you can see this is the molecular weight +1 of the brevetoxin. Here is the Delaware sample, and you can find that same molecular weight +1 in the Delaware sample. Okay. Another form of mass spectrometry, MSMS, looking for PBTX2, again, the same, finding that again. And finally, using something called mass spectroscopy, Nmr Spectroscopy, sorry, not mass spectroscopy, and we can then take a look at the structure, and taking a look at the brevetoxin standard, the Delaware sample, we end up with the structure of PBTX2. All right. The other things that you could do with this, obviously, growing this up in larger concentrations, we can look at this in a lot more detail now because this was only in a natural sample where the concentrations were perhaps minimal. In the cultures, we have found indications, and we are continuing to search in cultures for these toxins. We also looked at the effects of these toxins on other processes, like the hemolytic activity on cells, and we find that chloromorum does have hemolytic toxins. And so it has at least two kinds of toxins, if not more. So, but in any case, when we come to it, we end up with something that has a unique morphology, unlike any other species that we've seen described. Unique pigment, chloromorum, which is morum xanthan, which is now an indicator pigment. We have a molecular sequence that verified by secondary structure in standard statistical analysis that tells us it's in a category alone. It's got its own origin. It's really not being shared by others. All confirm the need to establish a new class, order, family, genus, and species. The pigment composition and molecular evidence separated from the xanthophytes and the rhaphidiophytes, the closest relatives. Okay, the closest relatives. And while some of the features suggest that it's close affinity to the rhaphidiophytes and the xanthophytes, this is not supported by any of the molecular evidence. And so it's in a class by itself. We have to basically erect a new class. And we're in the process of publishing this now. It's in review at this moment. And so this is a story. And you've been very, very patient to listen to all of this kind of chemical stuff. But it's to give you an idea that when you get an indication of something like this, that you need to follow it in a particular way. And it's the method that's involved here. That you look at it from more than one side, trying to confirm or deny any of the evidences that is tending you to go in a particular direction, and basically get to where you can support that conclusion and with your scientific colleagues. With that, I thank you very much for your patience, and I will be glad to answer any questions or explain anything that you may have about this, and we can go from there. Yes?
Did this lead to anything to do with the chicken waste?
The chicken waste. Well, that's my take on it. You know, from looking at the nutrients, I'm saying, well, it likes urea, and there's a lot of urea in that water. So it might be that that's one of the things that allowed it to bloom to the point that it did. Yes?
Have you also checked the groundwater by these chickens to see?
No, I haven't. No, we have to at some point kind of limit where we are. We're fortunate enough to be able to go as far as we did. Yes?
I'm just curious, as you tell this story and worked out this solution, how much time was expended? I mean, you took an hour to tell the story, but how long did it take you to come to all these?
It started in 2000, and we've sent in a publication last fall, and it's in review. So it's taken almost 10 years. It's longer than maybe some people like to have me work on it.
You said that as far as you know, this is unique to this Delaware situation.
Oh,
But that you,
I'm glad you mentioned that. Go ahead.
okay, I was just wondering if, assuming the chickens are involved, is there any place else with a similar morphology if you have, shallow water, warm water, and a high concentration of urine?
I'm glad you. I'm going to answer you in a second. I'm glad you mentioned this because it makes it sound as if Delaware is the only place where this thing is found. It's found in the New River. It's found in Florida, on the east coast of Florida. It was found in a Texas canal near Corpus Christi. It was found in Salton Sea, California. So this thing is really a lot more common. And the amazing thing is, if it was that common, why wasn't it seen before? It's truly a difficult organism to work with. But the whole answer of it is preserve samples. When you have groups that go out and sample, they can't spend the whole day looking at these things and every time they take a sample. So one of the first things they do is they'll take a water sample and put a preservative in it, like an iodine preservative or formalin, and all of this thing these things just shrivel up. Now, if you know what they turn into, you may be able to tell what these raisins are, but if you're not familiar with those, you're going to run into the same problem that my colleagues ran into and say, well, we can't identify these things.
Since these apparently cause fish kills, is there any, if there's any organization, agency, whatever, looking for similarities between these various places?
Oh yeah. Most of the natural resource facilities from each of the states look into these water quality issues. And here, for instance, in North Carolina, there's been a good deal of work done on an organism which was called Physteria. I don't know if you've ever heard of it. And that's one of the things that is like the ones that we experienced. Now, the interesting thing is they thought that this was due to Physteria, but we looked and looked and looked and could not find Physteria. And that's what took us onto this journey to figure out what the heck it is. Yes, please. No, if we were to compare it to the Florida red tide, it's probably a tenth as potent. So it could cause some problems, but I mean, it's not likely that it is. The thing where you'd want to watch is maybe shellfish, because the shellfish accumulate a lot of this material, and in digesting it, you get a lot of it in the hepatopancreas, which is the stomach of these. And so if you're eating oysters or clams, you might start seeing some symptoms. As far as those original symptoms that were explained, no one has ever really been able to confirm those stories. And whether they were just sort of figment of somebody's imagination or somebody looking for something to raise Cain about, we don't know. But we were never able to confirm that. Yes?
Do these blooms occur every year, or is it certain conditions during the year, during certain years that cause it? And what's the ground?
Yeah, they occur practically every year. And they're a summer species, so come June, July, you start looking for them and they start creeping up. It's usually the late summer where they really form large blooms, and sometimes they won't. If it's a particular rainy year or something that meteorologically different that particular year, it may not produce as many or as intense blooms. That's part of the mystery of a lot of this, is what kind of conditions do you need for them to really form dense blooms?
You only get a fish kill every year when they occur?
You can. Some years you don't. Yeah. It depends on how intense the blooms are. Yeah. Any other questions? Yes?
Is that Menhaden that you showed, is that the same as buffer C, or known as buffer C?
Exactly.
Which is usually much bigger,
Well, it's Brevoortia tyrannus, which is the pogey. That's the other name that they use for Menhaden. It's the Atlantic Menhaden, which is a migratory fish along the east coast. I mean, it comes in here too, and it routinely dies here too sometimes during the winter. Those are usually cold kills. Those are not toxin kills, fortunately. Yes?
With climate change and sea level rise, that type of thing, you said summer is when it would be we're going to have more and more summers. We can't tell it now, but the longer summers. Do we see a lot more fish kill in the future because of things like this?
That's a good question, and I think we're headed in that direction. What happens, obviously, is that as the water's warm, it just sort of expands the range that a lot of these species can occur in. And of course, as this particular organism is, it's kind of it's focused on in a summer environment, a warm, nutrient-rich environment, and that's likely to happen more often now. So we're likely to see that.
Last question.
Did I miss it, or did anyone analyze the Menhaden to see if you could find these organisms or what type of damage would occur and what's causing the kill?
No, that's a good question. No, it was just the fact that they were dead. But the thing that they did look for.
That's a good symptom.
Yeah, that usually is fatal, but no, the thing that they did notice about the Menhaden, and this was the thing that steered them away from Physteria, is that they did not have any of the typical lesions or open sores that they found with the Physteria kills. And so with this one, it apparently doesn't do the same kind of thing. It doesn't unnecessarily damage the skin and then open it up for infections.
Thanks, Carmen. My opportunity to just say one thing about this. In 2000, when this discovery was first made, or the first kind of indications were there, we started asking Carmen every couple of years, so how are you doing on this? Are you ready? Are you ready? Are you ready? And then this year, after we'd already scheduled all 4 of the talks, Carmen came up and said, you know, I'd like to be able to planet ocean someday. I said, really? Why? He said, I'm ready. So you can see it took a lot of time. This is really a testament to somebody willing to stick with a project. And he said it's over 10 years, it's 14 years. Okay. Publications are only now for review. So, Carm, you know what we do? We provide these wonderful little metal sculptures.
Oh.
You all know who does these for us?
You may go.
Megorum, right? The Megorum Jr. And I think he outdid himself this time. Now we're going to ask Carm in 5 seconds to tell us what the genus and species is. So here we go.
Oh, Scaniola extemorensis. Fantastic. Thank you.
For a great talk, Carm.
Thank you very much.
You notice that we're going to do tours tonight too. We're doing the reception over in the Marbionic building in the lobby, so you get a chance to see all of that. And then we have, let's see, Wendy Strangman Bassett. Wendy, raise your hand. Is going to, right there, is going to help with some tours tonight. Andrew Bordelais, way over there. Raise your hand. Thanks, Andrea. And did Bob get here tonight, Bob York?
He's in the lab.
Bob York's out in the lobby. So we'll have someone from my lab, someone from Carm's lab, and someone, or from Jeff's lab, and someone from Carm's lab, all to take you around the building. And the idea is to get around the building before we run out of wine. If we run out of wine, it's over. Okay, so thanks everyone for coming, and we're going to go over across the round circle to the Marbionic.
Hey Chaz, I'm getting used to seeing you in my